Hello, my name is Michelle Griffin. It's a pleasure to present our work entitled Single-Cell RNA Sequencing Reveals Fibroblast Heterogeneity Across Embryonic Origins of Skin. This work was carried out under the supervision of Dr. Michael Longacre. Skin healing following injury varies according to the location of the body. Facial skin wounds heal faster than back wounds. Hence, we take out sutures in the face after 5 to 7 days, and in the back we wait until 10 to 14 days. Fibroblasts play a vital role in determining the degree of wound repair following skin injury. However, what controls the specific regional phenotype of fibroblasts in response to skin is unknown. Lineage tracing experiments in mice have shown that dermal fibroblasts originate from different parts of the embryo. Fibroblasts across the body originate from 4 embryonic origins. The face is derived from the neural crest, the scalp from the cephalic mesoderm, the back from the somite, and the abdomen from the lateral plate mesoderm. Several studies have shown that embryonic origins of tissues may be important. Neural crest-derived cells are specific cell populations with multipotent and self-renewal properties. Neural crest-derived cells reside in various adult tissues. In response to injuries and stress, neural crest-derived cells have shown to be activated. For example, our lab has shown previously that the activation of neural crest stem cells following mandibular bone injury. So this led us to question whether the observed faster rate of healing in the face compared to the rest of the body may be due to fibroblasts being derived from a specific embryonic origin, the neural crest. And this led us to question whether the embryonic origins of fibroblasts govern the outcome of dermal scarring. To answer this question, we created a surgical mouse model to study the fibroblasts derived from the 4 embryonic origins. Within the same mouse model, we created 4 excisional wounds, 1 in each area of the body, including dorsum, abdomen, scalp, and face. At 14 days post-injury, we analysed the wounds by histology. To understand what may control the varied fibroblast response across the body, we isolated fibroblasts from the 4 embryonic origins using flow cytometry at 7 and 14 days post-injury and from unwounded tissue using the same surgical mouse model. We then analysed the fibroblasts using single-cell RNA sequencing to compare the gene transcriptome. So moving on to the results. H&E staining demonstrated that neural crest-derived wounds healed with thinner scars than the other 3 embryonic origins. Furthermore, Mason trichrome staining showed that neural crest-derived wounds had less collagen deposition than other 3 embryonic origins. So in summary, this data shows that skin scarring varies according to the embryonic origin. Having observed that facial wounds derived from the neural crest led to decreased scarring and fibrosis compared to the other embryonic origins, we hypothesized that we could improve dermal scarring by reprogramming fibroblasts to mimic neural crest-derived facial fibroblasts. Our single-cell RNA sequencing analysis showed that there were different fibroblast subpopulations in unwounded and wounded tissue. At day 14, there were 6 fibroblast clusters, or subpopulations. Looking at postdoctor 14, we wanted to see which subpopulation or cluster of fibroblasts may represent the neural crest-derived fibroblasts. And for cluster 2 was found to represent the neural crest-derived fibroblasts, as there was a significantly greater number of fibroblasts derived from the face in this cluster. And also, as we know that neural crest-derived cells have multipotent stem cell activity, we used the cytotrace program to see which cluster may represent cells in a least differentiated state, like stem cells. And this analysis also represented cluster 2. So in summary, cluster 2 represented the gene transcriptome of fibroblasts from the neural crest embryonic origin. So we then looked at the genes expressed by both facial and cluster 2 derived fibroblasts in depth. We found significant high levels of ROBO2 expression by facial and cluster 2 fibroblasts. We further confirmed that ROBO2 positive fibroblasts could be isolated from the face by flow cytometry, and there were higher levels in the face and following injury than from any other embryonic origin. So to understand the regenerative potential of these ROBO2 positive cells, we transplanted ROBO2 positive fibroblasts from the face by flow cytometry from GFP mice into dorsal wounds and compared the healing to wounds transplanted with ROBO2 negative fibroblasts. We found thinner wounds with less collagen deposition in wounds with ROBO2 positive fibroblasts. Collagen 1 protein and gene expression was also lower in wounds with ROBO2 positive fibroblasts than in wounds with ROBO2 negative fibroblasts, and to a similar level as facial wounds. Due to the improved healing of dorsal wounds with ROBO2 positive fibroblasts, we aim to improve ROBO2 signaling by activating ROBO2 fibroblasts using its ligand SLIT2. We treated dorsal mouse wounds with SLIT2 recombinant protein and analyzed the wounds at day 14 post-injury. Dorsal wounds with SLIT2 treatment showed decreased dermal thickness and collagen deposition compared to wounds without treatment. Furthermore, treatment of dorsal wounds with SLIT2 protein decreased protein antigen expression of collagen 1 and increased ROBO2 expression compared to wounds without treatment, and they were similar as facial wounds. So in conclusion, embryonic origins of fibroblasts do play an important role in determining their scarring activity. Neural crest-derived facial fibroblasts show a decreased scarring response. Neural crest fibroblasts show high levels of ROBO2 signaling. Activation of ROBO2 signaling by topical SLIT2 application decreases scarring in vivo. In the future, understanding the mechanism by which ROBO2 signaling decreases dermal fibrosis may provide therapeutic targets to overcome skin scarring.